Well, hello everyone. Welcome to this podcast for Biocore 383 on electron transport and oxidative phosphorylation. Before we begin our discussion, it's probably worth reminding ourselves where the events we've been discussing so far are occurring. You'll remember that pyruvate is produced through glycolysis, that's in the cytosol, and then that's translocated into the mitochondrion, where within the mitochondrial matrix the citric acid cycle occurs to generate reduced compounds, in particular NADH and a little bit of FADH2. We're now going to use those electron donors within the electron transport chain, and these reactions occur within the intermitochondrial membrane. The asymmetric release of hydrogen ions or protons into the intermembranous space is going to create a concentration difference across the intermitochondrial membrane, and it turns out this is going to be used to drive the formation of ATP during oxidative phosphorylation. Let's zoom in on the intermitochondrial membrane then and look at the arrangement of these electron carriers and where ATP synthesis is going to occur. All right, so here is the intermitochondrial membrane, and you can see that through the electron transfers of electron transport, protons are released at several key spots during this process. Those are boxed in light blue in this diagram. And you can see that those protons are released asymmetrically so that the intermembranous space becomes more acidic. It picks up additional protons compared to the matrix. Now remember that we were doing all of these redox reactions during the citric acid cycle to create NADH and FADH2 because these electron donors are a form of stored energy. And we said that we could relate these standard electrode potentials to standard free energy, and we've discussed how to do that. We also said that for NADH in particular, these redox reactions are a very good energy source, because if we can harness these electron transfers, we can liberate substantial free energy. In the case of NADH in particular, under standard conditions, that's minus 52.4 kilocalories per mole. And it's worth thinking about the strategy here that cells are adopting. Cells are taking NADH and oxidizing it and using the standard electrode potential to do work. And we can compare that to the energy liberated by ATP. That's minus 7.3 kilocalories per mole. So the standard free energy generated by oxidation of NADH, minus 52.4 kilocalories per mole, you can see that that can drive formation of a number of ATP molecules if we were to couple those reactions in some way. And that's precisely what aerobic respiration does. It takes all these small steps of electron transfers, storing energy in a concentration gradient of protons, and this allows for a substantial overall release of energy that can drive ATP production. So when you ask which is more efficient, going straight to ATP production, that's what happens, for example, in substrate level phosphorylation in glycolysis. That doesn't liberate nearly as much energy as this complicated series of electron transfers. So the next question is, how do we actually couple those electron transfers to ATP formation itself? And this subject is fascinating and has a long, rich history. In particular, Peter Mitchell developed an idea which at the time was thought to be rather crazy. In fact, at least urban legend has it, he did some of the experiments in his own basement. Mitchell suggested a way in which the electron transfers of electron transport could be coupled to ATP synthesis. And that's shown in the next slide. I want to point out that this slide doesn't show complex 2 where FADH2 comes in, but recognize that that's a component of the intermitochondrial membrane. But for clarity, this just shows complexes 1, 3, and 4, and shows the relationship between electron transport, proton release, and ATP synthesis. You can see in this diagram, starting on the left, that complex 1 receives 2 electrons from NADH, and it passes them through the complex. Eventually, those electrons are handed off to a mobile electron carrier, coenzyme Q, shown by the green oval on the left. Coenzyme Q then passes those electrons to complex 3 towards the center of the diagram. And as it does so, hydrogen ions are released into the intermembranous space, which is shown at the top of the diagram. Electrons are handed off to complex 3. They move through complex 3, and more protons are released asymmetrically into the intermembranous space. Another mobile electron carrier then hands electrons off from complex 3 to complex 4. That's called cytochrome C, and that's shown by the rust-colored oval in the center of the diagram. Cytochrome C then hands electrons off to complex 4. Electrons are moved through electron carriers within complex 4, and ultimately those electrons are transferred to oxygen, generating water at the bottom of the figure. Here again, some protons are released into the intermembranous space. So you can see that moving electrons through this system releases 10 hydrogen atoms into the intermembranous space. And now we're going to use those to generate ATP. You may remember that we can store energy in electrochemical gradient, and what's going to happen is that the protons are going to move through the large macromolecular complex on the right side of the figure, and this is the ATP synthase itself. This uses the energy from this proton gradient to generate ATP from ADP and inorganic phosphate. All right, so just a reminder, there are a couple of mobile electron carriers here, and they act as a relay system between the major complexes in the intermitochondrial membrane. Those are coenzyme Q, which hands off electrons between complexes 1 and 3, and cytochrome C, which hands electrons off between complexes 3 and 4. And again, complex 2, which involves FADH2, is not shown here. Now, one of the nice things about the new edition of World of the Cell is that we've provided some additional molecular realism to some of these diagrams. This is kind of the blobby version of the electron transport complexes in the intermitochondrial membrane. But here's a more realistic depiction of parts of 3 of these complexes based on X-ray crystallographic structures. You can see, if you look carefully at this diagram, the details are not something that you need to really be concerned with, except to say that it's worth looking at how the electrons are transferred. And you can see that the various redox reaction centers are coordinated in space in these complexes. So if you look at complex 1 off to the left, you can see that the iron-sulfur groups within some of the proteins within complex 1 are all positioned in a way that the electrons can be handed from one to the next in a very orderly fashion. And you can see that that starts at the bottom, sort of looks like the state of Florida, I guess, and you can pass electrons from NADH into the system. At the bottom, they work their way up the complex towards the membrane. Then ultimately they get passed off to coenzyme Q, the mobile carrier, which moves them to complex 3 in the center. And there again, you can see that there's a spatial coordination of all of the electron carriers within complex 3. Ultimately, this hands off to cytochrome C, a protein, but a mobile one, a peripheral membrane protein. Cytochrome C moves over to the right, as you can see, and transfers electrons into complex 4. They move through complex 4, and ultimately they're transferred onto oxygen to create water. A really beautiful, amazingly intricate system of electron transfers. Okay, so now let's consider in more detail what's going on with this whole proton gradient. And it was Mitchell's brilliant idea that you could somehow couple chemical and osmotic energy generated through this proton gradient to make ATP. And this interconversion of energy using an electrochemical gradient, because it involves osmotic and chemical energy, was called the chemiosmotic model for ATP synthesis. Now, you'll remember from our transport discussion that you can use ATP hydrolysis to do active transport. Well, it turns out that transporters, under the right conditions, can run that reaction in reverse. They are, after all, enzymes. And you remember that enzymes catalyze reactions, but where the equilibrium lies depends on the concentrations of reactants and products and prevailing conditions. And so that's true for any ion transporter. And so what we can do then is, if we have enough protons, we can drive this system in reverse and actually create ATP by passing protons through this channel. And that's exactly what happens. The energy stored in this proton gradient is often called a proton motive force, and that's really a voltage. So now you'll remember from our transport discussion that we can store free energy or expend free energy depending on the direction of movement of a charged solute. And we can relate that to the concentration of the solute inside and outside of a membrane, and to the voltage on the membrane and the charge of the solute. So we've already discussed that in our transport discussion. So we can use that equation to generate this proton motive force, or voltage. So we can divide by the Faraday constant to get the voltage. In the case of protons, Z, the charge on the ion is 1. And it turns out most of the time the pH difference is about 1 pH unit. And we can convert to log base 10. If you do all of that basic math, you get the following equation. The proton motive force is the membrane voltage plus a constant times RT times the change in pH divided by the Faraday constant. So it turns out that in this case, the membrane voltage is about 0.16 volts, and the change in pH leads to a voltage contribution of about 0.06 volts. Right, and again, these relate directly to the free energy calculations we did for ions already in our transport discussion. And you should be able to work with these sorts of numbers and massage them in the ways that we've just done. It's not hard, it involves just a little bit of algebra. So producing this ion gradient is a way to store free energy, which we can use to generate ATP. I want to remind you again of something, and that is that we've been discussing mitochondria, but aerobic bacteria can do all of these same reactions. It's worth thinking about where those events occur. Well, in bacteria, these occur at the plasma membrane. So in this case, a proton gradient is produced at the membrane, and there are ATP synthases embedded there, and they can be used to drive ATP synthesis in a very similar way to what we're talking about in the mitochondria. Okay, so far we've talked about how you can generate these kinds of proton gradients. They store free energy. Now let's talk about the evidence, experimental evidence, for how this works. Not shown in this figure. It's possible to biochemically isolate mitochondria in a way that preserves some of their membrane components. So you'll recall that the mitochondrion has an outer and an inner membrane. And it turns out that the proteins that synthesize ATP are embedded in the inner membrane with a particular portion of this multiprotein complex sticking out into the matrix. So in World of the Cell, these look sort of like lollipops. My son Christopher, who has autism, has cherry-flavored Tootsie Pops every day as part of his meals. And so I know a lot about lollipops, and maybe that's why I think of this as the lollipop model for ATP synthesis. So you can see those lollipops protruding into the matrix in part A in this diagram. So in an intact mitochondrion, we can measure the extent of electron transport by giving an artificial electron donor and measuring an output. There's a way to do this biochemically. The details are not something that we need to concern ourselves with. And if you measure whether electron transport occurs under those conditions, in an intact mitochondrion, of course, it does occur. Moreover, we know mitochondria produce ATP, and so you can provide ADP and inorganic phosphate and appropriate reagents and perform similar biochemical assays to show, of course, that an intact mitochondria in the presence of oxygen, or in the presence of an electron donor and an acceptor that can drive electron transport, you will get ATP synthesis. So that makes sense. We can do electron transport and we can synthesize ATP. But now here's where it gets interesting. So we can biochemically disrupt mitochondria in such a way that we can isolate their membranes. And you can see that we get little spheres of membrane that represent the intermitochondrial membrane in part B. All right, so in this case, we can assay for electron transport and ATP synthesis, and you'll see in this case that we can do both. We can do ATP synthesis and we can do electron transport. And the reason that it runs in this direction is because we can create a proton gradient in the middle of these membranes, and this can be used to drive ATP synthesis. More on that in a moment. All right, so these lollipops are actually the F1 subunit of ATP synthase. We'll look at the detailed structure in a minute, but this is the part of the ATP synthase that is not in the intermitochondrial membrane. Right, again, these spheres are intermitochondrial membranes, and what we were able to show with this experiment is that you can reconstitute in miniature what's going on in the intermitochondrial membrane and the intramembranous space in these little vesicles. Right, so now it becomes possible to strip off those lollipops and look at the membranes only, the membranes with the lollipops, or the lollipops themselves. So let's look at that. Right, if we take these particles and we dissociate the lollipops, the proteins, from the membranes, we can show that electron transport still occurs. But now, interestingly, ATP synthesis does not. However, remember that ATP synthesis and the utilization of ATP to move ions through channels are reversible reactions, and it turns out in this case, now you don't drive ATP synthesis. Instead, if you give the system ATP, it can catalyze the hydrolysis of ATP to ADP plus inorganic phosphate. And what that indicates is that the lollipops can still do their ATPase activity. They still have enzymatic activity, but because they're no longer in the membrane, it is not coupled to electron transport. Conversely, the membranes can do electron transport because it's not coupled through a proton gradient to ATP formation, we don't get ATP synthesis. All right, well, we can continue this kind of process. So the membranes isolated on their own, shown on the left, they can do electron transport, but because the ATP synthase component is missing, the lollipops, we cannot make ATP, nor can we hydrolyze ATP, the reverse reaction. That's because the lollipops are what do that. Conversely, the lollipops alone can't do electron transport. That's shown on the right. They can't do ATP synthesis either, and the reason for that is that now, because electron transport and the proton gradient that's produced as a result is not coupled to ATP synthesis. Conversely, the lollipops can, if given ATP, hydrolyze it to ADP and inorganic phosphate. That's because they carry the enzymatic activity that's normally associated with ATP synthesis. And it turns out we can do the full Monty. We can put all of these together, reconstitute particles, and now we reconstitute both electron transport and, given the appropriate ion concentration, ATP synthesis. So to review, if we have membranes only, we get electron transport. The lollipops only can hydrolyze ATP to ADP because they contain the enzyme, but it's not coupled to a proton gradient, so they can't make ATP. But if we put them both together, the lollipops and the membranes, then we can do electron transport and couple that to ATP synthesis. Really cool experiments. Right, now I told you that UW was at the forefront of some of the metabolic studies, including iron-sulfur proteins. It turns out another UW scientist was at the heart of figuring out in detail how ATP synthesis works in the system. And that's Paul Boyer. Paul Boyer and John Walker developed something called the binding change model. Paul Boyer was a professor at UW-Madison and won a Nobel Prize for this work. All right, we'll talk about that in a minute, but before I do that, it's probably worth saying something. ATP synthase, the purple molecule on the right in this diagram that we've already discussed, is a channel. Protons move through the inner mitochondrial membrane through this channel. So the purple thing really is a channel. But it's worth pointing out something. These electron transport complexes are not channels. So although protons are released from these channels, this is through a complicated series of electron handoffs and redox reactions within each of the complexes. And the protons are actually released asymmetrically, but they don't pass all the way through these brown complexes. So the brown complexes don't act as little pipes through the inner mitochondrial membrane. The ATP synthase does, however. So this one is a channel, and protons move through it through what's called the FO subunit. Right now, you'll get some opportunity to think about how electron transport and ATP synthesis are coupled. I just want to preview that by saying that one of the things that blocks this process is cyanide, and in fact, at the early part of the 20th century, chemists were developing ways to perturb electron transport and oxidative phosphorylation, and there's a really rich history there. It's kind of the beginnings of forensic science and kind of think early 20th century version of CSI. And actually, someone who lives across the street from me, Deborah Blum, wrote a really engaging book called The Poisoner's Handbook about this rich history. It's a great book. Encourage you to read it. Deborah's a really great person. But we'll give you more opportunity to think about that later. All right, now let's return to this ATP synthase. This is the machine that couples proton movement to conversion of ADP, from ADP to ATP. And there are two main parts. Each goes by the capital letter F. F stands for coupling factor. The one through which the protons move is called FO, and that O, it's not a zero, it's an O, stands for oligomycin sensitive. Oligomycin is an antibiotic which blocks the function of this subunit. The F1 subunit down below, that's the lollipop part. That's the part that protrudes into the matrix. And it turns out that that thing is, in an amazing way, is going to rotate. And that rotation is going to generate ATP through this binding change model that Boyer and Walker developed. So in the membrane, we've got several parts. There's a static part that doesn't move. That's the proton channel. That's the A subunit. And then you can see that there's kind of a shaft labeled with the letter B that connects to the F1 part of the ATP synthase. And then there are 10 C subunits. Well, the numbers vary slightly from organism to organism. And only one of these interacts with the A subunit at any given time. Right in the matrix, there's something called delta. The delta subunit connects to the B stalk, so the delta part is at the very bottom there, doesn't move. And then there are alternating alpha and beta subunits connected to a gamma subunit. The gamma subunit is like a crankshaft and it turns, as do the alpha and beta subunits. And there's a little piece called epsilon that interacts with the FO subunit. So we've got static parts, and then we've got this whole rotating thing, and it's the rotation that creates ATP. And this is where Boyer and Walker's work comes in. So if we look down onto the F1 subunit, or we look up from the bottom, that's what's shown in the next figure. And you can see how this works. So ADP and inorganic phosphate bind to a beta subunit, which is in the open conformation. That's the capital letter O on the upper left of this diagram. Proton moves through the FO subunit, and this causes rotation of this whole complex by 120 degrees. As this rotation occurs, you can see that the beta subunit labeled with a capital letter L, which had been holding an ADP and an inorganic phosphate, now catalyzes the formation of ATP. That's the happy sunshiny molecule on the upper right. At the same time, the subunit that was unoccupied and labeled with a capital letter O now becomes labeled with a capital letter L, because it has now bound another pair of ADP and inorganic phosphate. All right, you'll notice that the subunit that has catalyzed the formation of ATP is labeled T for tight. It has bound these very tightly and catalyzed that reaction. The next cycle of rotation, that ATP is released, so that now the subunit that was in the tight conformation and had just created the ATP releases its ATP, becomes in the open conformation, it's colored red, and O for open. And we can keep doing this rotation. You keep cycling through unoccupied beta subunits binding a new ADP and an inorganic phosphate. There's a rotation, tight conformation, creates ATP, another rotation, it releases its ATP, and there's this revolving door of beta subunits binding ADP and inorganic phosphate and ultimately creating ATP. An amazing, remarkable machine. Now, let's consider what's going on in this binding pocket. When one of these beta subunits enters the tight conformation, it dramatically affects the free energy of required to synthesize ATP. Now you remember that the standard reaction in solution for ADP going to ATP is plus 7.3 kilocalories per mole. If we compare that, though, to what's going on in the ATP synthase, we can see that delta G in this case is about 0. So all of this has allowed for an energetically favorable situation that allows for much more rapid and ready formation of ATP. All right, so what is kind of amazing about this is the prediction is that you've got to actually move this ATP synthase and rotate these subunits to create this cyclical change in binding of beta subunits and the way that they interact with ADP and inorganic phosphate, creating ATP, releasing it, picking up a new ADP and inorganic phosphate. So what if there were a way to actually visualize the rotation of an ATP synthase? Well, it turns out that there is, and biophysicists have done the following experiment. We can take actin, and we can use a molecular glue, basically, called avidin, and we can connect that to the FO subunit. We can then actually glue the bottom part, the F1 part, down using a particular set of conditions to immobilize it. And if that actin filament is fluorescent, that thing that's glued to the FO subunit, normally the FO and F1 rotate relative to one another. The FO is embedded in the membrane, you'll recall. But if we fix the bottom, then the top will rotate. And we can look at the propeller of motion of this fluorescent actin molecule. So let's do that. And here's the result. Pretty striking. So what we're watching here are fluorescent actin molecules, and the short ones are on the right, medium in the middle, and long ones on the left, and they rotate at different velocities. But you can see clearly they are whipping around, and this is direct evidence that this ATP synthase works via rotation of its subunits. This is an amazing result, an amazing achievement of biophysics. Right, you can't really see much in these movies except that the rotation's occurring. Here's a molecular model that summarizes what we believe to be how this works based on crystal structures. You can see that the FO subunit is up at the top, embedded in the membrane, and the F1 subunit, the lollipop part, is protruding down into the matrix. So let's just run this movie forward. So what you can see from this is that the beta and alpha subunits are stationary, and the FO subunits, the C portion of the FO subunit is rotating, and it's connected via the shaft. And you can see that as this rotation occurs, it's displacing and affecting the alpha and beta subunits down below. The beta subunits, the orange ones, kind of the yellowish orange ones, are the ones that catalyze the production of ATP. So the rotation of the ATP synthase, the central portion of it, is what catalyzes, through this binding change model that we've been discussing, conformational changes in the beta subunits, which then can catalyze synthesis of ATP. And the ATP molecules are the kind of the magenta things that you can see flying out of this complex. This is one of the most important macromolecular machines for life as we know it on this planet. And I hope you appreciate how amazing the discovery process was that led to our current understanding of how all of this works. I hope this has been helpful. Thanks for listening. We'll talk to you again soon.